Welcome to MOOC course on Introduction to Proteogenomics. This is Dr. D.R. Mani's second lecture. In the first lecture, he had discussed about various strategies employed for data normalization. In today's lecture, we are going to continue the discussion about data analysis and especially the batch correction and missing value imputation. These two things are very important for any type of omic data analysis. Just because you can generate large amount of data set in a very short time using mass spectrometry or NGS platform, that does not mean that the data quality is very high. You have to be very cautious, very clear that what data you are analyzing and make sure that you do the proper ways of normalization, batch correction as well as the missing value imputation before you start further data processing. In this way, the batch correction removes the technical differences in the data, whereas missing values need to be imputed to get the better outcome. Sometimes you have to take a call. If there are many missing values in your data set, then probably that is not a good data. You need to trash the data and it starts all over. Or let us say if you have seen only very few places the data points are missing, but rest of the data is there, then software can utilize some resources, some ways of averaging and imputes the values to fill out those missing values. Again, there is lot of statistics and considerations required. What should be the way to do the missing value imputation, but that is what Dr. Mani is going to talk to you today in his lecture. He will also explain and talk to you about batch correction methods like lemma and combat. On the other hand, he will discuss about the missing value imputation, which is one of the very important considerations in the big data analysis. So, let us welcome Dr. Mani again for his lecture on batch correction and missing values imputation.
So, the next topic in this set of slides is batch correction. So, we just talked about normalization where we are trying to make all the samples similar. So, I don't know how many of you have heard of batch correction, just a few. So, batch correction is something you apply when you think you have different batches of experiments you did and you think there might be a difference between those batches. So, you have 15 TMT experiments you need to do. You did 5 in January, 5 in May and the remaining 5 in winter. And this is all one project and you had lot of vacation left over, so you weren't there to finish all of them. So, now you want to put all of them together and do an analysis. So, it is likely that the data you get from each set of 5 are going to be very different or significantly different. And that is because of technical variation, not because of biological differences in the sets of samples you used. So, correcting for that is called batch correction. So, let us say you have a scenario where you are looking at breast cancer, you have ER positive, ER negative and HER2 positive samples. So, you run all your ER positive samples in January, you run all your HER2 positive samples in May and then you run all your PR positive samples in December. And now you have 3 batches again which have differences. And there is big differences between what you did in January and what you did in May. Is it because of the difference between ER and PR or is it because of the batch? Answer? Yes, you can't tell because the way you designed your experiment, we can't deconvolve whether it was because of the batch or whether it was because of biological differences. So, that is why statisticians say you always have to run a randomized subset when you are splitting things into batches. So, if you had ER, PR and HER2 positive samples, in January you pick a random subset of ER, PR and HER2 positive samples, you put them in your 510 plexes and run it. You do the same in May and you do the same in December. Now, if there is a big difference between January and May, you know because it is because of your batch, because there are ER, PR and HER2 positive samples in both sets and it is not a biological difference. So, that is why when you run experiments, you generally tend to design them so that you lay them out and you randomize the samples to the degree possible. So if 3 people are working on a project, you don't want one person to be working on ER positive samples, another to be working on PR positive samples and so forth. You want each one to work on a random subset of all the samples. So any distinction that is of importance to you in the biology, that has to be randomized. If you don't, if you have batch effects, you cannot separate the batch effects from the biology. And if you did your experiment correctly and there is a batch effect, then how do you deal with it? And that's kind of what I'm going to talk about in the next few slides. So what I say, like normalization, batch correction attempts to remove technical artifacts in the data. But normalization is usually on a sample by sample basis, whereas batch correction is on a sample subset basis. So you have a group of samples that you ran at some point that needs, that resulted in some technical difference between some other group. So batch correction can be used to remove systematic technical differences. So different operators, different time of running it, different machines, different column you used for your LC, things like that. But as long as you have normalized, so you have randomized your data, you are in good shape. So you decided to run all your ER positive samples in the beginning of your study. You are doing everything in 1 week, but you decided to run all your ER samples on the first 3 days, and then you are doing your PR samples on the next 3 days. And on day 2.5, your column got clogged and you had to throw it away. So now all your ER samples were run with the old column and most of your PR samples were run with the second column. There is a difference. Is it the column or is it biology? So you can't tell. But if you are alternating ER and PR samples and there is a big difference, you know it's your column. So that's the importance of like randomizing your analysis when you are doing sample processing and experimental analysis. So the thing is, if you have an internal, if you have a reference that you ran and you try to use that to minimize the difference, it would be okay for a pilot project, but if you try to publish it, the reviewers will not accept it. So you have to have a randomized study and then do some sort of batch correction. You can help if you have at least same thing you ran across your study, you can maybe try to use it to do better batch correction, but it's not ideal. It's better than nothing, but it's not the ideal approach. If the question is whether a protein was different between type A and type B, so like cancer versus normal, a protein is different, you do it 3 times and you see the same difference. So in that case, you probably don't need batch correction because all you're doing is ratios of cancer to normal in the same batch. But if you are now, if you have 3 replicates, your statistics will be much more robust. So you can combine all the 3 replicates to find out which proteins are different. In that case, if there is a big difference between the same protein measurement in your different runs, then your statistics will fail because there is way too much variation, your p-values will be very high. So in that case, it will help to have batch correction. So it depends on how you analyze the data. If you are looking at each replicate separately, drawing some conclusion and checking that conclusion across, you don't need batch correction. But if you are combining all your replicates and doing a unified statistical analysis, then hopefully you would have randomized your replicates, but if you didn't, you would do batch correction. You have only 10 samples that you can put on a TMT 10-plex, and then you submitted it and the reviewer came back and said run it 2 more times. So then you can run it 2 more times, you don't need a reference. But if you are doing a study which requires 15 TMT 10-plexes just to do one replicate of the study, then you would have a reference because you have to link the different 10-plexes. So if I think the rule of thumb is if your experiment will fit in one TMT reaction or one TMT experiment, you don't need a reference. But if it is going to span more than one, you should think about having a reference. In some cases, if there are like 2 or 3 10-plexes, depending on the conditions and the experiment, maybe you don't need it. But if it is a big discovery study where you have 100 samples and you need to do like biomarker discovery or proteomic analysis of all those samples, then you would have a reference. So what are methods we have for batch correction? I have a couple of methods listed. I won't go into too much detail here, but 2 common ones used are Lima is a package that is used for a lot of analysis. You can also use it for like differential marker analysis and so forth. It builds a linear, like a two-way ANOVA model, and then uses that to do the batch correction. And there is a R package called lemma, and it has a function called remove batch effect. I won't go into the theory or details here, but you can explore that, and I think they have some examples and stuff if you want to take a look at it. Another option is called combat. So this is a empirical base estimation of how to do the batch correction. So Bayesian analysis usually tends to be more robust than traditional frequentist analysis. So this kind of uses a little more robust method, but this also has mechanisms that make it robust. It uses what is called moderation, which is essentially another way of doing empirical Bayesian analysis. So both these tools are relatively useful, and you can try both. They have differences that might make it more appropriate for one project or not. So suppose you have two batches, but one batch is more sacred than the other one. So in your batch correction, you want to take the second batch and make it similar to the first one, instead of just putting the two together and correcting however. So if you want to do that, I think combat will let you do that, but lemma will not let you do that. So there are differences like that that might dictate which you want to use, but both are reasonable to take a look at. So I think I have made this point multiple times. So batch correction can obviously introduce artifacts. So because of that, I would not do batch correction unless there is proof that batch correction is necessary. So you look at something and you say, oh, this set is very different from that set. If you can show that, then you would go and do batch correction. Otherwise, batch correction can introduce artifacts that might look like signal, that might get rid of signal. So you can get a lot of false positives and false negatives if you indiscriminately use batch correction. And one example showing here is what I was saying. The experiment was not properly randomized, and so one batch has more of one type of things than the other batch, and when you do batch correction, it looks like there is a differential signal, because batch correction says, oh, this batch was one, this batch was a second batch, and I need to go correct for both. And when you do that, the differences introduced could look like biological signal. So here is an example that Karsten worked on in our group. So we had a experiment where there was RNA-seq done from about 105 samples. So those are the red samples. So you can see this is plotting the expression value for all the genes measured using RNA-seq. This is a box plot. So you can see the first batch was all fine. There were one sample that was slightly low, but most of them were fine. If you look at the second batch, this one was new sets of samples, but it also had replicates from this set. So things marked with a star underneath are replicates from this batch. So you take the same sample from this batch and you run it like a year later. You can see how different they are. And so the question is, now I want to put these two together and do an analysis, because I got more samples. When you have more samples, you get more statistical power. And so how do I do it now? And so for that, this is a case where you show there is a batch effect, and without correcting the batch effect, you can't do the analysis. So we do go and correct it. And once you correct, they all look similar. And so here is an example of how that looks similar. So the left side shows hierarchical clustering. Hierarchical clustering groups similar things together. So when you say, here are all my samples, show me things that are similar and put them together. You can see all the blue ones, which are the new batch, grouped together. All the red ones, which is the old batch, grouped together. And the green things in the middle are the replicates. So even the replicates don't go together, they go with the batch. So this is a very strong batch effect that overrides any biological signal you might have. And so when you correct it, you can see now that the reds and the blues are all mixed up, and the greens actually line up. The replicates line up with each other. So now after batch correction, a sample and its replicate are the most similar, which is the correct place to be, not with the things in the same batch. So we say that after batch correction, the batch effect has been removed, and now you can do the analysis. So that's kind of an example of where we used it. The next topic is missing values. This one I have a lot of slides, but I think many of them are technical and probably unnecessary. So I'll just zip through it quickly. So mass spectrometry is prone to more missing values than RNA-seq or most of the genomics methodologies. So in like 10-15 years ago, if you used an Affymetrix microarray to measure a sample, you would get a measurement for every single gene. There would basically be no missing values. But in proteomics, it's not possible because proteomics is stochastic. You are measuring things that fly in a mass spectrometer, and what flies once may not fly again. What flies in one sample may not fly for another sample. And so you tend to see a lot more missing values in proteomics. To make this worse, when you're doing phosphoproteomics, which is really the interesting part of proteomics, because you can look at signaling and kinases and how they work, the phosphosites may be present in one sample and completely absent in another. So maybe some pathway is activated in a subset of your cancer samples, but is not activated in all your other samples. So the phosphosites that represent the activation of that pathway will be present in like some small subset of your samples, but not present in all your other samples. And so you get a lot more missing values in phosphoproteomics. When you try to do statistical and machine learning analysis, many of the tools need all the data to work. If you have missing values, the algorithm cannot be applied. And so the question is, how do you deal with missing values? So in statistical theory, there are 3 types of missing values in increasing order of, I guess, a worry index. If you are in the first one, you don't need to worry about it. You can throw away the data and you'll be fine. So that is called missing completely at random. So randomness is very important. If your data is missing in such a way that the things that are missing are completely random, then they're not going to affect your biology. And so you can say, okay, fine, I don't care, I'm going to throw it away. I have 200 proteins that are completely missing at random, then I can just ignore it. The second part, missing at random, is that the missing value depends on some part of the observed data. So for an example here is peptide intensity is missing based on characteristics of the peptide. So if the peptide has a specific amino acid, it's missing. So here, so if the peptide has a specific amino acid, at random times it's missing, it's not always missing. So there you know the characteristic, and once you know the characteristic, then the missingness is random, because which sample it's missing is random. But if the sample has a specific amino acid in that peptide, then it's likely that it might be missing. But whether it's actually missing or not is random. So in that case, it's called missing at random. So these things you have to be a little more careful. You can't just like throw it away. But the most worrisome part is missing not at random. And unfortunately, all proteomics data, most proteomics data is missing not at random. Missing not at random means the missing value depends on the missing data. In other words, it's missing when the value is some specific thing. If your intensity is less than some count, the data is missing. So that is missing not at random. And so that is the most worrisome type of data. And there you have to be very careful on how you deal with it. You can't just like casually throw it away. But it's also the hardest to deal with even with statistical theory. So I'll just go through like a pragmatic approach to how to deal with this. I will not go into all the details of what we do. We don't need that. So here are some approaches that people use to deal with missing data. So one is called complete case analysis. So this is, you throw away any data that is missing and then you do your analysis. So if a protein was observed in 95% of your samples but missing in the 5%, you throw it away and then you do your analysis. So this loses a lot of data. And in phosphoproteomics, this may not be a good thing because you will lose a lot of your signaling peptides. You may lose a lot of your biomarkers too, even in the proteomics data. So you want some other method. So one way is throwing away things that have a large proportion of missing values. So that may be reasonable. So in other words, if you have something that is missing in more than 80% of your data, you can say that's way too much, I don't want to deal with it. So if a protein is missing in more than 80% of your data, you don't look at it. That makes sense because statistically there's not much you can derive from looking at a small set, and also you don't know how it's different in the others, and so it may be okay to drop variables that are missing. So in the analysis we do, we have a relatively high threshold. I think if the data is missing in more than 70% of the samples, then we throw that away. So we take a protein, if that protein has not been observed in 70% or more of the samples, we throw it away. So if we start with 12,000 proteins and we apply that filter, about a few hundred, the maximum 1,000 proteins will get thrown away. So it's about like 5 to 10% of proteins generally fall into that category. If you look at phosphoproteomics, the number is larger, it's more like 25-30%, but still it's not that big a number, and it's a reasonable thing to do in most situations. So the other thing to do is impute the missing data. So you can say, okay, I don't want to throw away things that have missing data. My analysis method needs the data, so what can I do? So one thing you can do is you can say, I'm going to carefully figure out what the value could have been or should have been, and then fill it in. So that's called imputation. So you can impute missing data and then complete your data set. For that, there are several methods. The best ones use some sort of a machine learning model. So they look at all your data, they look at things that have been observed for the thing that is missing, and then they kind of predict the missing value should have been this by looking at the entire data set. So it builds a machine learning model by looking at the entire data set and then comes up with a imputed value for data that's missing. So we have found that there is this method called k nearest neighbor imputation that works reasonably well. There are a couple of other machine learning based methods that also work well. So if you really have to fill in, so the way we do our analysis, we start with the full table, we apply the threshold. If it is missing in too many samples, we just throw it away, and then for the remaining, we use a k nearest neighbor imputation to fill it in, to do the analysis. There are some analysis methods that can actually deal with missing data. In those cases, we don't impute, but if you are using a method that cannot deal with missing data, we impute using KNN or some machine learning based method. So before you do a marker analysis, you should not use the information of the classes for making any decision, because if you do that, then you are biasing your analysis to the groups that you know, and you should not do that. So you would look at the entire data set. Let's say your perfect biomarker is present in your cancer and not in the controls. And so you have 50% cancer, 50% controls. If you set this threshold to be 40%, you will throw away all your biomarkers. So you would set the number to be high enough. So let's say you set it to be 70 or 80%. So then you retain things that are present in only a few of the cancers, but are missing in all the normals and maybe even a few cancers. Then you will keep those markers. When you do k nearest neighbor implementation, remember it's called k nearest neighbor. So what it does is it's going to say, okay, I need to fill in this protein for this sample. I'm going to find samples that are similar and find what values they have and then fill it in. So it's going to look at other control samples and then fill it in. But if it was missing in all the control samples, then the imputed value will not be that clear. But if some had some small values because of noise or something like that, then it will just average the noise and create a value that will fall into the noise. But if it was missing in the tumors, then when you look for similar samples, it will find more tumors. And then it will fill in with a value that is specific to the tumors. So this algorithm will actually correctly take group into account without knowing about the group. It doesn't have to be actual technical replicates, but you need biological replicates for the same group. Yeah. If you had 2 samples and it was missing in one, there's not much you can do to fill it in the other. But if you had 100 samples and it was missing in 5, you can use the other 95 to fill it in.
So, I hope today you have learnt about what is batch correction and how to perform this kind of analysis. You also learned the important strategies of batch correction, which is lemma. The design matrix is used to describe comparisons between the samples. For example, the treatment effects, which should not be removed. The function in effect fits a linear model to the data, which includes both batches and regular treatments, then removes the component due to the batch effect. Another method for batch correction is combat, which is robust as it can do a model based adjustment to remove the artifacts. The batch correction also need to be done correctly, otherwise a wrong strategy may lead you to the artifacts. So, next important thing is the missing value imputation, which you have heard. This is one of the common facts people see in the various experiments, especially the mass spectrometry based data generation, whereas sometime some values you can't see for every single protein, and what should be the considerations to impute these missing values is very important. Again, as I mentioned in the beginning, that you need to ensure that missing values are not too much in your data set, especially not more than 70 to 80%, otherwise the data is not real. You should try to not use the data set at all. If there are only very few missing data points, then you can utilize the missing value imputation strategy to try to recover that information. There are different type of missing values, like missing not at random MNAR, or missing at random MAR, as well as missing completely at random or MCAR, of which the last one MCAR is commonly used for the proteomic data set analysis. You also heard the k nearest neighbor model based imputation, which is a preferred statistical analysis with missing values when compared to the mean or median population. We will continue our discussion about different strategies employed for data analysis, and the lecture will be continued by Dr. D. R. Mani in the next lecture. Thank you.